Tonight we're going to look at different processes that what we're doing through formulation with Haripada, Stefan Schmidt from the side of a CMO, Wayne Froland, who works in different aspects with Merck, and Joanne Beck. And what we want to look at, we're all trying to do our best in our jobs, trying to look at what technologies, strategies. Cost cutting is always something everyone asks for, meeting needs and utilizing creativity, whether it be people or databases, and lessons learned. And I'd like to start with Joanne. From your presentation tonight, what has been a key tool since you go from start to finish? What has been a key tool for your team in success? Is it databases? Is it management? How do you manage a project since you're from A to Z?
Well, we have, program management and project management. I think the key to, be successful is, well, you manage the project, make decisions that are crisp and early and timely. And then in terms of successfully developing a process, I really do believe that the principles of quality by design are the key to success.
Thank you.
You can't skip too many steps there.
And Haripada, she mentioned tonight that you just reformulate, and that it's a simple formulation. It probably takes, like a second or two? I'd like your perspective, since you're on the formulation for Lilly. What goes into a drug product, formulation, reformulation? What is something that we should look at, whether we're research or PD or bioprocess, some considerations that you might have in mind?
Actually, I'm in a formulation group, but my core responsibility is biophysical characterization. So as far as formulation is concerned in Lilly, New Jersey side, we do not have a formulation for different stages like phase 1, phase 2, phase 3. We do not have from the very beginning, we try to understand the molecule as detailed as possible, and we do not have a pre-formulation, late stage formulation. We develop one formulation, and we have been successful for many different molecules. We have not failed for a single molecule. And so the key is to understand the molecule very well and develop the formulation at the beginning itself so that you don't have to change the formulation at different stages. So that is our approach at Lilly, New Jersey side.
And how does formulation, we look at it, we are a global society, and how do you deal with regulations through EU, the US, and Asia? Is there something that you work together with teams to deal with different issues?
No, for formulation development, the basic thing is that it doesn't depend on the agency, which agency it is the formulation, as long as your molecule is stable and it meet your expected shelf life, that's it. So, and here in formulation, we try to first apply the basic biophysical tools to understand the molecule, and which gives the proper direction to move forward. But one of the key things in this particular approach is the correct and use of these different biophysical tools, and there are a lot of interpretation or misinterpretation of the data. So that really, as far as my group is concerned, that we pay a great deal of attention on formulation development. And there, when we do that, in most of the cases, our early direction and the final result matches very well. For example, there are concepts that, when I say the concept and misconcept of the biophysical data, like we say that, okay, TAM of a protein, given protein in condition A is higher than condition B. But when we do a accelerated stability, we see that it doesn't match. And then you say, oh, okay, this particular DSC or some other method didn't tell. It is really not the proper way to judge this data. So the way we do, not only we look at the biophysical data in detail, like TM, for example, is just a thermal stability, conferential stability, but the molecule has both thermodynamic as well as kinetic stability, how they are correlated. So all these things we look at before we make a decision. So that's why so far we have whatever the early direction we get by basic biophysical tools, we have a very good success correlating with the accelerated stability and getting a quick answer in a short period of time.
Thank you. Is there a quick trick for us in research for viscosity and stability? Things, say, 2 simple assays or something that we could add to our toolbox while we're looking at early stage molecules? Something that you would suggest?
For the development you're talking?
Yes.
Oh, okay. For the development, as far as techniques are concerned, and I would certainly recommend DSC is a first thing that you have to do. And dynamic light scattering is something very important, where you can get a very high throughput way, some interaction parameter, which is very nice. And then, which I pay a great deal of attention of reversibility. That actually we also apply, we recently applied for heat-induced viral inactivation for process development purposes. So you can get a temperature, for melting temperature for a given molecule, some, let's say 50 degree or 60 degree, and other molecule we got little higher, let's say. But that doesn't tell everything. So the thing is what it says, how reversible it is. So that reversibility parameter we also look at. So if you combine the thermal reversibility, differential scanning calorimetry, and dynamic light scattering, and the fluorescence, intrinsic fluorescence, and that probably would give you a very good direction which way you would go.
Thank you, Haripada. Wayne, you have a dearth of experience taking things through and working with Merck. Question for you is, how have you been successful with CMOs? What have been troubles and what have been successes throughout your career?
So I would say trouble comes in 2 forms. In terms of your preparedness and readiness to actually outsource a program. I think Joanne hit it when she says that you can do a lot of outsourcing and you can use a wide range of vendors, but you can't really outsource your core competency. You do need that. You must have that. You must understand the program before you can tech transfer. The second is making bad decisions about where you're going to put it. So good due diligence is absolutely critical. There are 3 areas that are absolutely important. Quality systems, you need to understand, because you're working with another company's quality system, you're not in your own. This is, of course, something your quality organization needs to understand also. From a technical point of view, CMOs, for the most part, are very good, and they know what they can do really, really well. But they're also it's very important to them to also understand, some of the aspects of the technology you may be bringing that are on the edge, and that you have that honest discussion, so that you really have your eyes open when it comes to tech transfer. The other part is the business, and that, when I say business, I mean communication. And this is on both sides. The ability to do efficient tech transfer and get the product in and have a reasonable relationship, whether it's going to be for the phase 2 or phase 1, or we're talking phase 3, perhaps we're even talking a commercial contract. The ability to make decisions and to have good communication within the sponsor company, we can react to things, but if something is happening at the CMO from a tech transfer or from a manufacturing issue, and you need to have a decision, you need to have a decision fairly quickly. You can't go back to the sponsor company or us in this case and work our way through our normal processes. So you have to have a process for how you're going to interact with your contract manufacturing partners. It's not fee for service, not with biologics, and I'm not saying in a very joining way talking about all, including small molecules, but drug substance manufacturer and contract manufacturing organizations, it's a partnership, and you need to have that kind of relationship where you can make decisions and make decisions quickly. I think the final point can be only one point of communication. That is for each of the three technical areas. The business piece has to run smoothly, the technical piece has to run smoothly, and the quality piece has to run smoothly. There should be just those three individuals, one team, one point of contact in terms of the organizations. That way communication channels are never blurred and decisions are made quickly.
Thank you, Wayne. So what you're saying is when Merck or yourself and a team looks to outsource something, you bring three people that lead that project, or is there one project manager and
Merck
three?
Will bring 30. But within that 30, no, it's not quite that simple. But
yes.
What I'm talking about is actual communication. There has to be someone in the quality organization, in a contracting manufacturing organization that has direct contact with somebody in our quality organization. Same goes for the technical aspect. There needs to be one technical lead. There certainly is a team behind, because if you're talking commercial, there's a lot of work that needs to be done, and we don't have 2 years. We've got to move fairly quickly to get to the PPQs, because it's very, very expensive. So we do need to have teams working, but when it comes down to, are we going to do this or are we going to do this, they know where to go.
Thank you. And on the other side, Stefan, we all know that we bring you perfect projects, and that everything we bring you is a fully formulated protein or antibody that works first. What is something that would help you as an outsourcing CMO, and something that you find that could help the process along in your ideas? What could we do?
I think it's very interesting what my partner to the left side just mentioned. I'll probably start with the last point about communication, and I think communication definitely is key, and from our side as a CMO, it's also very important to be able to match and understand the expectations, because expectations can vary to some degree. Also depending on the level or the stage where the project comes into our place. I mentioned in the morning when I was speaking, we can accept in principle anything from a gene to deliver drug substance in the end in vials, but we can also have projects coming in between, which means a transfer, tech transfer. And tech transfer is much more complicated the later the stages where you are, because you mentioned the gap or the balance or disbalance sometimes of quality organizations, and that is what we're seeing all the time. We have a full variety of clients, from very small startup companies up to very, very experienced large pharma corporations, and they all have different expectations in quality terms. In some cases, we are the ones sort of teaching our clients what quality should mean. In other cases, we are kind of educated on what other large corporations think is quality needs, and we have to sort of manage the gap in between with our own quality system, which is definitely good and solid, and we have had very many successes in the past. But I think this is one thing to manage all the time, the communication and managing expectations. And what would help us in all cases, and no matter what the clients are, really if the customer has a clear idea what he wants from us, which timelines, what kind of experience of the molecule exists. And there we see also some gaps in between. People coming with molecules they haven't fully understood, and we find out during the transfer that, okay, there is something with the glycosylation which the partner hasn't seen before. Just an example. But really, communication is the key point of all success in CMO and outsourcing.
Thank you, Stefan. The question is, you said that they don't have fully vetted projects. My question to you is, there 10 items that maybe someone with a molecule should come to you and say, hey, look, I've looked at solubility, I looked at X, Y, Z. What are things that you would like that would maybe make a tech transfer easier?
That's, again, the Santa Claus wish list, 10 items, a lot, really. It would be
Find 5 for today, and Friday, everyone at 5 can listen to Stefan for the rest.
For sure. For us, timelines is very important to know what kind of connected things are going on in the background, meaning what kind of patient acquisition is running on, what kind of timelines you're having for submission. This is one of the key elements to sort of time it also on our side in a good way. Then the other thing is also, the later you are, the more the volume expectations increase, that people have a really clear understanding for which stage, what amount of material they require, because sometimes they come, let's have one batch that should be sufficient, and we got a call a couple of weeks later, oh, we forgot there's another batch we need. So this interaction from the start to have a clear understanding what is your process, what kind of things come up in the near future, this would help us a lot. And I think this is probably the two main things I would say. And of course, then later on, all the details about the molecule itself, about the cell line, and I think you can name a longer list than just 10 items, for example. But I think coming back to the first two things is very important, timelines and amount.
Can I ask you a question? Sure. Stefan, so do you find that a lot of your sponsors or clients, whatever, come to you with very unrealistic timelines?
Definitely. They believe, I have a project here, I'm a big company and you can do it very fast. And we can do it very fast, but we have also our own timelines. And a simple example is the supply chain is not to be ignored, because we have a full range of, let's say, disposable reactors. In principle, we have all the big manufacturers in our house that we can deal with, but you have, since our size is not as big as Boehringer or Lonza, for example, you have a different service level with the suppliers. So that means requiring something very unorthodox would take us longer to get in than if, for example, Lonza has it in their stocks already, for example. Yeah, this is something you take into account.
Yeah.
Joanne, from the other side, since you hand off projects, what are things that you make sure that you hand off with? What are some things that you help to communicate with your CMOs about?
Yeah, I mean, I agree with the comments both Wayne and Stefan made. I think we need to understand our product. We need to understand pipelines, timelines, and communicate those timelines. Communication is extremely important. Having a point of contact, I think, one point of contact, not, you know, that doesn't mean that it's a team of one that goes to, but it's the one point of contact, like a project manager or whatever, on both sides that they communicate. One thing that I think is important to us, and we've had a tough time, it's tough for us to outsource biologics, is the flexibility of the manufacturing floor. We don't do monoclonal antibodies, and a lot of things are set up, a lot of companies are set up to do the very traditional 3-step process with a viral filtration and a UFTF in the end. We don't have a lot of places we can go to do that can do perfusion. But, you know, that's one of the things that I would like to have more available, and see how we can have more flexibility in terms of the different options for processing.
And Wayne, have you seen also with different molecules, maybe made by Pichia, maybe made by different yeast, like we see with Novartis, have you seen for outside the norm, just the land of CHO, is there different CMOs that can really handle, you know, multiple different products that come out of different cell lines?
Yes, absolutely. You need to know what the skill sets are for the contract manufacturing organizations you're going to be working with, and there are experts out there, really profoundly good ones, maybe not at super high volumes. Sandoz is probably, when they were in the business, was probably the bigger tanks for the prokaryotic systems. But they're out there. Certainly, they're out there, right? And we can access those. That is there. It's just a matter of you need to go in and understand, either by first recognizing from reputation, but then measuring and getting in a discussion and really understanding whether or not the CMO has the expertise. Because it's been my experience, a couple things, and I don't want to go back and give a non-answer to the previous question too. It's been my experience more than once. When we bring a manufacturing process into a contract manufacturing organization, and in one case, this was an E. coli-based program, the contract manufacturing organization actually does a hell of a lot better job than we could have in the house. Look, they're running all the time. They're running multiple programs. They've got people coming with different ideas all the time. They get almost like the regulators, they know a little bit more than we do, even as sponsors. And so, and in E. coli, had a very recent experience where the contract manufacturing organization, this is a CHO-based product, is actually much more efficient, better, and I think even to a certain extent, a higher quality product coming from the contract manufacturing organization that we would be able to generate at that same scale. So I think from deciding who you're going to work with is super critical. The relationship, it's a business relationship. Contract manufacturing is a business. They have a margin. It's a nice margin, right? And they need to make that. And you have to realize that when you're outsourcing, that you're going to pay a certain amount of money to have them do your work. And hopefully, what you get back is really high quality work. So you have a contract. You got to work through that. Sometimes that's fun. Sometimes that's not. But at the end, you get it done. You know exactly what the terms and conditions are. Then there's two other pieces that are very, very critical as a company who's outsourcing. You got to know. One is the timeline, and for sure we're going to go back and accelerate it. The second piece is scope. You can't really outsource something if you don't know what it is you're going to outsource. You have to line it up. Now expectations are there. Everybody knows what we have to do, and that goes right down to the analytical methods. That goes right down to the item specifications, how we're going to manage raw materials, what's going to be the overcharge. What do you call that when you float that 7%, 9% on top of the raw materials?
Oh, yeah.
Extra happy money.
Handling fee? Yeah. You need to know these things and understand these things. You have a good defined scope. Now you start with a very, very clean understanding of what each company is expecting in terms of expectations and what we can go in return, because believe me, it's going to get blurred anyway as the program accelerates. And they do. They all need to accelerate these days.
Yeah.
Joanne, any thoughts on that?
Oh, no, I thought it was interesting, the comment that you made that, you got higher quality product out of a very good CMO. Yeah, I think also, when we deal with our internal manufacturing or our pilot plant or our internal manufacturing, we can become abusive sometimes in that, we change things at the last minute. Oh, we don't like this raw material, let's get this other raw material. Oh, no, don't do it this way, do it that way. And so I think it's a kind of a surprise when you go to a very disciplined organization like a contract manufacturing organization, and they want you to follow their systematic approach to things. And it's kind of a wake-up call, I think. And it's a good wake-up call. I think it builds discipline in the PD organization to understand the product and the process and be very systematic in doing tech transfers. And I think it will improve by outsourcing more products. We will improve our internal tech transfer success rate.
And question, Joanne, with predicated history from R&D to process to maybe stage 1, how do you educate your outsourcing partners? What do you give them that you can show a history of the molecule so that they can see what you've done and where you've come from and what may have been tried before?
Yeah, so I mean, we have created, we've been very disciplined in terms of building on the QBD principle. In other words, starting very early on, we have certain templates that we use. We fill out, what we understand about the product, what we understand about, hotspots of the product, and then whatever we have learned about our process and the process controls, et cetera. So we've been using, tech transfer templates that we've built, and any technical reports that have been written in the process of developing the process, we hand them over. So we are very open in terms of what we know and we don't know about our process.
Thank you. One last question for everyone. I'll start with you, Haripada. What is it that we could do better for our colleagues that would help you in what you do for formulations?
As I said,
or biochemical characterization.
In my view is that it is really, really important to use a technique correctly. There are many cases which I think that there are interpretation is, there is a conflict, how things are being used. So if you use the techniques in a correct way, you really can get a very wealth of information with very minimum experiment. And that is the key. And like, for example, in case of one product, we had some low pH viral inactivation. There was issue, there was aggregation and all. So when it came to our biophysical group for resolving this issue, so the conventional method, what you'll do, okay, do some DSC, do some other stability, and look at and see where in different conditions where the molecule will be stable. But we didn't do that way. When I looked at this thing, so we designed, and because some of these cases, the design of the experiment needs to be innovative. So that's why you need to have a thorough understanding of the technique and thorough understanding of the system very well. So in that case, what I applied, simple refolding, unfolding kinetics, I applied over there, and it solved the problem in a very short period of time. This is not a common thing, like where you want to apply those things. So it really depends on case to case. So that kind of things, it's kind of, we try to, it is not that everywhere you can use a common method and get a answer that you need. Specifically for biophysical analysis, that's what I experienced.
Okay. Thank you. And Wayne, is there something that you would say that from your perspective that we could, as scientists, help for the outsourcing, something that you've learned?
Outsourcing is partnering with someone to do the work that you know and understand. So I think Joanne started it, and if you don't know and understand what it is you're going to transfer, you're not ready to tech transfer it into a CMO. You need to have an organization that can do tech transfers. It's a learning process that's, albeit a painful process, but you do need to be able to tech transfer, and you do need to know what it is that you're going to transfer out. And before you actually enter into this, you should know and have an organization within your company that is able to do that. That can start in research, that starts in early, we outsource very early for phase 1, phase 2 sometimes. But certainly when you're getting to outsourcing a phase 3 or outsourcing a commercial product, you have to prepare yourself first.
Thank you, Wayne. And Stefan, is there one thing that you'd like to add?
I think outsourcing is, if you take an example, it's a bit like leaving your baby to daycare. So you have your own child. This is you have invested a lot of effort to have this project being done. Your baby, then you leave it to someone else taking care of it, and in the end you want to have it back because in the end you want to bring it to the market and want to treat patients. So all what I've heard now is a lot around matter of trust. You have to establish a certain level of trust. It's the same if you choose a kindergarten, you want to look first how is the atmosphere there, how they're dealing other kids, is there a lot of shouting, fighting, is there a clear structure, how is the headmaster of this kindergarten running? So quality systems, it's a bit similar to a CMO actually. Yeah? And then on the other hand, you see a CMO in the kindergarten treats many children simultaneously. So they have a huge experience accumulated with different clients, different parents, different characters of children, and they all more or less get them successful through kindergarten and later to school. School for me would be something similar to on the market, having it in real life in the end. Yeah? And this is really, as I said it before, it's really around establishing trust, reliability, and interacting in a proper way together for the better, greater good of this life of patients in the end. Yeah.
Thank you, Stefan.
I think I'd like to keep the babies in the house, but how about a couple 13-year-old boys?
The
Then we're not talking about kindergarten anymore. Yeah.
Anyway, Joanne, if I could ask you the same question, just one thought of something that we could maybe improve.
I don't know. I'll throw out something completely different here. I think analytics is something that we've had a really, really hard time transferring. And I think we need to do a better job of understanding analytical methods and even transferring a simple analytical method like size exclusion chromatography. We have differences between the different labs, and it's hard to identify whether it's the actual HPLC, let's say, or it's the resin. We buy the same column from the same manufacturer, but it behaves differently. And I think, that's my thought for the day, I guess, is that analytical methods are a real tough thing to transfer. And I was wondering if you guys have the same kind of experience, actually. Especially, with formulation. I mean, those are very, I don't know if you're outsourcing anything at all like that, but I find it's tough to find contract manufacturers that are also very good at analytical methods. And we have to go to specialty labs sometimes.
Well, one of the issues is an LCMS, a triple quad, a QTOF, an Orbi trap versus an SciEx, and this all give different mass spectrometry to look at and shapes, and all the units are different. They all are maybe a mass spec, but they all give readings differently. You look at a mass spec of 6500 AB SciEx versus an Orbi trap versus an Agilent QTOF. You put the sample on the same machine, and you'll get 3 different answers. And that's one of the issues with analytics. You have to standardize the machinery, just like the bioprocess. You have to standardize what you're looking at. Thank you so much for your time.